So I tried to sort of put two sort of disjoint pieces of work together into this talk, and you'll see how they relate, hopefully. So first, I want to talk about compressing information that lives on a de Bruijn graph, and then I want to talk about compressing, in a sense, paths through that graph. So let me start with this setup. The motivation here, some of you may be familiar with it, is large-scale sequence indexing. So the goal here is to index large collections of sequences. So there's a lot of work sort of since the dawn of bioinformatics in indexing reference sequences. A relatively recent trend is indexing raw data. And there's a lot of reasons you might want to do this. One common example I like to throw out there is, imagine that you discover a gene that hasn't been annotated before and it seems new. And now you want to go back through existing sequencing data and see if it's been observed before, right? If it's been expressed, say, in RNA-seq data. So there's many reasons why you'd want to do this. How you would go about it is somewhat of a harder question and a very open one, I would say. There's a very nice review. A number of the co-authors are present here that covers, sort of the different approaches people have taken so far to this problem. I'm going to focus here on what are called color aggregative methods. And I'm going to introduce this by way of a very simple example. And this is actually sort of the approach we took in our first pass at this problem in an index called Mantis. So imagine you have some collection of input experiments. We're going to first represent each input experiment by its set of K-mers. So nominally here, we're losing some information. We're discarding relationships among strings of length greater than K, and we're not considering frequency here. I'll mention you could consider these things with frequency, but I'm looking at the most basic setting here. And we're going to take the set of input experiments and we're going to represent it by the following kind of map. We have a set of keys, which is the union of all of the K-mers that appear in any experiment. And then we have the values associated with each key is just simply an occurrence vector, right? This is a bit vector that's 0 if this K-mer occurred in this experiment and 1 otherwise. So it turns out that this representation is useful, but there's something you can do to vastly reduce the size of the storage, and that's to recognize that many K-mers have exactly the same pattern of co-occurrence. And so this is, in fact, what we did in our first approach to this problem in an index called Mantis, where we build a key value store based off of the counting quotient filter data structure. It's actually, we use an invertible hash function, so it's exact. The key is the union of all K-mers, and each key, instead of now mapping directly to some large bit vector, maps to an identifier, and that identifier signifies or relates to a particular occurrence vector, right? And we can play some clever tricks here, like compressing the bit vectors themselves to make them take less space using something like RRR compression. And we can also arrange the color IDs so that the most frequent ones are the ones that are smallest in magnitude, something like Huffman encoding. And this actually provides very fast query, and it scales well up to thousands of experiments or input samples. But the problem is we really want to index tens or hundreds of thousands of experiments, maybe even millions eventually. And a key observation we had in trying to think about scalability here is the K-mers grow at worst linearly. If I add a new experiment, I'm adding at most, a number of K-mers is linear in the size of that experiment. But the color classes grow super linearly because when I add new K-mers, I add both rows to this index, right? I add more keys, and I also increase the number of columns, right? The number of experiments that I have to represent co-occurrence patterns over. So the argument here is that we need a fundamentally better encoding for color classes. And we thought about this for a while, and one thing that sort of jumped out at us is the reason the initial encoding worked of indirection from looking to represent the vectors directly to representing identifiers and grouping all of the K-mers that have the same pattern of occurrence together is that there's many exactly repeated patterns. But this isn't unique to exactly repeated patterns. There's many approximately repeated patterns as well, right? And so one thing you can think about doing is referentially storing or referentially encoding this information, trying to find K-mers that are close, K-mers whose occurrence vector is close to the pattern of many other occurrence vectors, and storing just the representative plus some deltas. So the key ideas here are we want to exploit coherence between the rows of this color class matrix. And the other idea is that if we know a K-mer's color vector, we generally know a lot of information about the color vectors of its neighbors. So here's an idea that we originally had, and much later, after sort of trying to implement this, we realized it's essentially the same as this idea was presented in this 1991 paper by Buchstein et al. In the information retrieval literature. But the idea is we have this collection of bit vectors, which are our occurrence vectors. We're going to build a graph over them. So for simplicity, consider the complete graph, where the edge connecting two endpoints represents the Hamming weight between those vectors. So how many bits we would have to flip to get from one to the other. And then we want to take the MST of this thing. Right, and I'll talk in a little bit about how we can use this to decode efficiently the individual vectors. So here is the MST of this particular graph of Hamming weights. Unfortunately, there's a number of problems. First, there's just many color classes. So building the complete graph is a non-starter. It's too big. Even if we don't materialize it, just computing all of the edges is prohibitive. And these are really high dimensional and sparse vectors generally. And so we tried some schemes like locality sensitive hashing to try to find neighbors efficiently, and that didn't seem to work for us in practice. I'm not claiming it never would work, or maybe we just didn't try the right scheme, but we didn't find one that worked. So we had this idea instead. What if we only consider the possibility of adding edges between color vectors that are adjacent on the de Bruijn graph? So what I mean by this is if two K-mers are adjacent in the de Bruijn graph, we consider if they have the same color vector. If they have a different color vector, then we consider adding this edge to our graph, the graph that we're going to take the MST of. So here's an example of this. I'm just going to show a couple of examples of how we would find an edge. So these two K-mers are adjacent. So we compute the Hamming weight of the two bit vectors, and we insert this edge into our overall graph, our color class graph. We see the same thing again. This is a different edge. By the time we're done visiting all of the adjacency relationships in the de Bruijn graph, we have a graph that looks like this, which is in general a subgraph of the full color class graph. So this is already an approximation to what we want. We're not guaranteed to get a minimum weight spanning tree of the full graph, but we find that we get something quite nice, right? This actually finds a lot of the low weight relationships. And in reality, all we really need is a low weight spanner, right? We don't need a globally minimum spanning tree to get a good compression. So this is just showing the difference on the top between the MST derived from our graph, which is the subgraph that is only manifest in the de Bruijn graph, versus the complete color class graph, right? So it's a higher weight spanner, but not by much. And so how do you decode this kind of information? Well, the first thing we do is we just add an all zeros vector to guarantee one connected MST for all of the individual bit vectors. And then we use the following encoding. We store each bit vector once, each unique bit vector, and we don't store each unique bit vector once. Conceptually, we have each unique bit vector as a vertex of this MST, but what we actually store are just along each edge from a parent to a child, the delta, right? The bits whose parity we would have to flip to reconstruct the child node from the parent, right? And now if we want to reconstruct a particular node in this graph, all we have to do, starting from that node, is walk up the tree from the node we start at to the root, flipping the bits that we encounter along each edge, right? So if we wanted to construct, for example, this red vector that's all ones, we first flip 1 and 3. That gets us to this blue vector. From here, we flip 2 and 4. That gets us to this green vector. From here, we flip 0. At that point, we'll have flipped bits 0, 1, 2, 3, 4. It's all of the bits here, right? So this turns out to work really well in practice. So what I'm showing here is if we take random sets of RNA-seq samples from 200 up to 10,000, and we build this representation on them, how much space these representations take up on disk, which is essentially the same as they take up in memory. It's a very straightforward encoding. And so what I'm showing here is the red dotted line is the size of the key value map that maps a K-mer to its ID. So that's sort of equal across both representations. The blue line is the size of storing the RRR compressed bit vectors. Now, again, we're collapsing the exact redundancy. So this is just the unique bit vectors being stored as RRR compressed bit vectors. And then the green line is what we get when we store all the values of this MST. And the thing to sort of interesting to look at is if we look at the size of the MST divided by the size of the RRR representation, we can see that as we add more and more experiments, the ratio actually gets better, right? The compression gets better. So once we get to 10,000 experiments, it's less than 10% of the size of the original representation. So this does a pretty good job of compressing this color information. There's some other tricks that I don't have really time to mention about how you make it fast, like building a cache, but it's pretty efficient. And it's worth mentioning that there are some other very related ideas that I think have some nice complementary relationships to this. So there's a series of work that is sort of outlined in these papers here. This second paper recently appeared on bioRxiv. I believe it's accepted to Recom 22, that exploits a similar idea, but does it in a different space. So here we built this color class graph, which exists independently of the De Bruijn graph, and then found a low weight spanner of it. But you could also imagine just doing referential encoding directly on the De Bruijn graph, right? And this idea is very interesting. It has some benefits. For example, there's no longer a need to store a label for each node. I don't have to store an ID associated with each K-mer. I can just store the delta of its signal from its predecessor or from whatever I'm representing it with respect to. But the challenge here, or the potential thing that you could gain, is it's restricted to local referential encoding, right? I can't take two K-mers that exist in a completely different part of the graph and then represent one color class encoding with respect to another. So I think there's maybe some synergy here that could be exploited. So the second topic I want to cover is nominally a very different approach, and is very much ongoing work. So I don't really have results to show, but I want to explain the idea because I think it may be a useful one. And this has to do with reference indexing using colored compacted De Bruijn graphs. So luckily, the speakers who came before me in this afternoon session have told everyone about De Bruijn graphs. So I don't have to spend much time talking about those. I'll just mention what the colored and compacted adjectives refer to here. So if we think of a De Bruijn graph as a graph representing the sequence in a set of reads or a reference genome, we can think of a colored De Bruijn graph as the graph representing the sequences in a collection of such genomes or collection of such read sets. So here I have two references, the blue and the orange. The top graph represents the De Bruijn graph with a value of k equals 3. And here it's colored. Every node is associated with the reference string in which its K-mer occurs. And the compacted colored De Bruijn graph here is just the condensation of this graph so that all non-branching paths are represented by a single node, right? So here, all paths with where nodes have in degree of 1 and out degree of 1, except for the first and last, are condensed into a single node. So the motivation for this is that there's a number of tools, and I'd say argue a growing number of tools that use the colored compacted De Bruijn graph as a reference index. And what I mean by that is as a way to represent many different reference sequences and then do things like map or align reads to them, or pseudo align reads to them, or compare them in interesting ways based off of alignment or mapping. And so the idea that I'm going to talk about sort of builds off of work that we previously did in an index called Pufferfish. It's not really too important to know the minute details of how Pufferfish works, but I do have to give an overview of the index. So the idea behind Pufferfish is we're going to build the compacted De Bruijn graph on a collection of reference sequences, and we're going to store a couple of useful pieces of information. So first of all, we need to represent all of the K-mers. The way we're going to do that is by taking all of the unitigs of the compacted De Bruijn graph. These are just these non-branching paths here, and we're going to concatenate them together into one array, this 2-bit encoded that we call the C-seq array, or the contig sequence array. We want to be able to find the boundaries of these unitigs because we're concatenating them all together. So we do this with a simple boundary vector. If our boundary vector supports rank and select, that gives us useful operations to sort of perform the indexing that we care about. And so the way the index works is like this. I want to know if a K-mer occurs in my set of references, And if it does, I want to know what references it occurs in and the positions in those references where the K-mer occurs. So the way that the query works here is you give me a K-mer. The K-mer is queried in a minimal perfect hash function. That hash function points to some offset array that gives me the position of that K-mer in the concatenated unitig array. So by default, the minimum perfect hash function only is collision free over the space of keys you build it on. But here, since we're storing the key set directly in this contig sequence array, we can just verify if the K-mer we queried with was in fact the one we found. If it's not, the key was not there, the K-mer didn't occur in the reference. If we find it, we're good to go. I can tell you what contig it occurs on by issuing a rank operation on the position here, and I can tell you its relative offset of this K-mer within its unitig by issuing a select operation on that rank. So that gives me the index of the unitig where the K-mer appears and the offset of the K-mer in the unitig. Now we have one more element of the index, which we call the contig table, and that just stores a list of triplets for every contig, and the triplets tell me what reference the contig occurs in, the position it occurs at on that reference, and the orientation. And a contig can occur multiple times on the same reference. So this is just a list of triplets that enumerate all of the occurrences of this contig. All right. And so when we think about the size of this index, the first thing that really dominated the size when we built it on large sequences is this position table. This gets pretty big. Each cell in this position table is log of C-seq length in terms of number of bits. But we came up with a mechanism to reduce that significantly, essentially by sampling only some K-mers to keep their positions and reconstructing the positions of others. For lack of time, I'm not going to go into details of that. But once we eliminate that bottleneck, it actually turns out that when we build this on large collections of sequences, the next bottleneck is this contig table. That is storing the paths in the graph that represent the original references becomes the bottleneck. So here, what this graph is showing is we built the pufferfish index, the sparse index, over all of bacterial, fungal, archaeal, and viral genomes in RefSeq from about a year ago. And the index itself takes about 170 gigs. And if we look at only the components that are over a few gigabytes in size, the dominant component is this contig table, right? This thing that stores the paths. So the question is, how can we compress this, right? So the problem we're interested in solving is compressing what I'll call reference-tilings. That is, I want to know a given reference is represented by a sequence of unitigs that tile it, and I want to represent that information in small space in a way that's still efficient to access. So the naive encoding that we currently use is very straightforward. Every contig has a list of references, positions, and orientations where it occurs. And those are stored independently. So to store the reference where a unitig occurs, I need log of n bits, where n is the number of references I'm building my index over. To store the position within that reference, I need log L bits, where L is the maximum length of a reference I'm representing. And then we can steal just 1 bit for the orientation. And so the question we had is, can we do the same thing that we did for the position information for the tiling information? That is, can we store this information just for some unitigs and deterministically reconstruct it for all the others? So it turns out that it's really useful to think about tilings if you want to think about how you might solve this problem. So a tiling is just, we're representing each reference as the sequence of unitigs that spell it out. So here there are 3 references. The first one consists of this, the first unitig followed by another unitig followed by a third. The second reference consists of 6 unitigs. And this is sort of the representation we have. This is implicitly encoded in the contig table. And what we'd like to do is to instead store the information for only some of those, here are the ones that are highlighted in black, and reconstruct it deterministically the ones that we haven't stored the information for. Okay, okay, so when we think about reference tilings, here's the key idea that we have. So on a tiling, if I have the information stored for one unitig, I can get the information for the unitig that comes before it, the preceding unitig, by knowing just the information of what is the preceding nucleotide, right? And the reason this works is because a unitig decomposition ensures that all K-mers are unique, occur exactly once in the unitig decomposition. So if I know what nucleotide occurs before the current unitig, that completely determines the terminal K-mer of the preceding unitig, which completely determines the preceding unitig. Okay, so the idea is this. For a sampled unitig, I'm going to store the reference position, the reference, the position, and the orientation. And for a unitig that's not sampled, I'm just going to store the predecessor nucleotide. And if to reconstruct the position information, I walk the predecessors until I see a sampled unitig. That yields the reference, position, and orientation information. And then since we know the lengths of all the unitigs, we can just walk back and reconstruct the position of the unitig that wasn't sampled. There's some details that are hairy that for time I'm not going to go into, like orientation, but you can handle that as well. So the key idea is on a tiling, we can traverse from one unitig to the one before it by prepending the correct preceding nucleotide to the first K-1 mer. The most important thing to think about here is unitig occurrences. And each reference is tiled by a sequence of unitigs. The last component we need is that a unitig can appear many times over the collection of references. And so instead of thinking just about the unitig itself, we're going to think about each occurrence of the unitig. And we're going to assign each unitig a rank, which is its the number of its occurrence in this reference. And the claim we have is there is a one-to-one function between the rank of a unitig, UI, R, and the occurrence information, which is the reference, position, and orientation where it occurs. This is a complicated way of saying that if I told you what the unitig is and what occurrence of the unitig this is, that would completely determine where it appears in this reference tile. That's all this is saying. And so turns out one problem we have with the previous approach, which is that though telling you the preceding nucleotide tells you the contig that occurs before, a unitig can occur with many different preceding unitigs, right? Different references can spell out different paths using the same unitigs. So for example, here, the red unitig is first preceded by the blue one, in the second reference preceded by the gray one, and in the third reference preceded by the blue one. So what we actually need to encode is the pairing, the unitig-unitig pairing between a unitig and its predecessor as it occurs across all ranks of that unit. So the solution to this is what we're calling a handshake. And the idea is that previously we said we were going to store just for the sampled unitigs, the reference, position, and orientation, and for the non-sampled ones, the predecessor nucleotide. The information we're going to add is we're also going to store for each unitig a successor nucleotide, which tells you what is the first K-mer, what is the kth nucleotide in the unitig that occurs after me. So that is the first nucleotide not contained in the current unitig. And now the claim is if a unitig has its predecessor nucleotide, and that predecessor has a successor that agrees with the current unitig, then we're good to go, right? Then this uniquely determines this particular pair of occurrences in the reference. So now the claim is that by walking predecessors and checking these handshakes until a sampled unitig yields the information that's sampled, we can reconstruct the position of the starting unitig. I guess the important thing is what is the scheme for holding this information? So we propose that there's a predecessor array such that Pi at J contains the nucleotide P, where P concatenated with the last K-mer of unitig I is the last K-mer of the unitig that precedes the jth occurrence of UI and R. And the successor array is just the array such that Sij contains a nucleotide S, where the first K-mer, the last K-mer concatenated with S is the first K-mer of the unitig that succeeds the jth occurrence of U and R. The idea here, and this is sort of the last slide I have, I realize I'm almost out of time here, is the way the algorithm works is given UI and R, that is a unitig, I I know I'm out of time. Given a unitig UI and R, which is its rank, we just want to know the unitig uj that precedes it and the rank of that unitig. That allows us to do this recursively. So given uj, we can look up that unitig using the Pufferfish index by prepending the nucleotide we need to prepend. We get the rank of that preceding unitig. Then we select the one that agrees with this handshake, meaning the preceding one that has that nucleotide in that position, whose successor has the nucleotide that our unitig has, and then we return this pair. There's a lot of argument, there's a lot to be done in the proof that this works. We've actually sort of worked this out, that you can do this and store this information. You don't have to worry about things like loops in the graph. And that all follows from the fact that we're encoding the reference tiling as it occurs in the references and not the graph itself. But we believe this encoding works, right? And so the only thing, there's a lot of open questions here, like what is a good strategy to sample unitig information? We could sample just the first unitig in each reference, but that could lead to really long reconstruction time. So you want some fixed distance sampling scheme. And the optimal way to do that is not clear, and it's probably hard, but we haven't convinced ourselves necessarily of that yet. So in closing, I'd like to thank all of my collaborators on this work, both on the ongoing work on reference tiling compression and on the work on color class compression as done in Mantis. Thank you.